Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speakers, Patrick Neuvenhausen, Director, Senior Consultant, Quality Management and Compliance, and Gerardo Gomez, PhD, Director, Service Line Lead, Quality Management and Compliance, both from Pharmalex.
Well, thanks for the introduction, Alex. It's a pleasure to have the opportunity to speak to Bioprocess International, the great audience you have, so looking forward to it. So if we kick it off, I would like to show you the agenda for today, what we're going to talk about. And the first point is regulations. Then we take a bit of a deeper dive in facility design. Then we go a bit more in qualification and commission, and we end with the takeaway points. So I would like to now hand over to my dear colleague, Gerardo, and we start off with the regulations.
Thank you so much, Patrick. Hi, everyone. Gerardo Gomez here. Very happy to be joining these Bioprocess International Ask the Expert webinar, and very happy and honored to be joining my colleague here, Patrick Neuvenhausen. Let me start, jump right into our topic of discussion. Let's start with the regulations. We'll be talking about regulations from the two main or most addressable regulatory agencies, FDAs and EMA. I'll start with the FDA 21 CFR Part 211. It's Part C, talks about buildings and facilities. This is very, very focused and very much explicit about the need to be attentive to how your buildings are designed. It must be of suitable size, construction, and location to facilitate cleaning, maintenance, and proper operations. And the second bullet really reinforces that. It talks about space. So small is troublesome. Too large is also trouble because you will require a lot more of what we'll be talking more in depth, utilities, air, etc. So proper size is important. Flow of components, drug product containers, closures, labeling, everything around the flow of people and materials is key. And operations should be properly specified and performed within specifically predefined areas, each of adequate size. There should be separate or defined areas or such other control systems for the firm's operations as are necessary to prevent contaminations or mix-ups during the course of the procedures delineated in that particular section of the CFR. So everything there that I just mentioned is just highlighting that. And next, that's a code of federal regulations. The important aspects there, we're going into the specifics, is lighting. Not only lighting, for example, for visual inspection, that's almost intuitively and goes without saying, but lighting for everything. How do you make in a packaging line, for example? You do your line clearances. There's poor lighting. How are you going to make sure that the line clearance is done correctly? So as seemingly trivial as that, the agency expect for us to be attentive to lighting in general. Ventilation, air filtration, air heating, that's your HVAC, is a big component of your facility design, which is the topic of our talk today. Sewage and refuse, washing and toilet facilities, sanitation or decontamination, and of course, maintenance. Ben, let me just scroll down quickly for the entire slide. along the same lines and the same subpart C, additional guidings specifically for aseptic processing. It's floors, walls, and ceilings must be smooth, hard, so they're easily cleanable. So smooth and soft will not make it. Hard, good, but porous will not make it. Temperature, humidity controls, top parameters for even more critical for aseptic processing. Air supply filter through your HEPAs, high efficiency particulate air filters. There's your HEPAs and ULPAs under positive pressure, of course, regardless of whether the flow is laminar or non-laminar. A system for monitoring your environmental conditions, that's when they said a system, is your EM program in general. And a system for cleaning and disinfecting the room and equipment to produce aseptic conditions. Last point there, a system for maintaining any equipment used to control the aseptic conditions. That's your overall maintenance program, engineering, calibration, and maintenance program. So that's FDA. For Hydrolex, Patrick, back to you.
And thanks for outlining that, Gerardo. And of course, besides the US FDA guidelines, we also have some European guidelines, and they are more or less pretty similar. They are high level, but you need to interpret that how you can make that work for your facility. Important to know is that when you look at the Hydrolex Volume 4, Chapter 3, about premises and equipment, it talks a lot about quality risk management. And again, that your premises, they should be preferably laid out in a logical order corresponding to the sequence of your operation. And if you go further about premises and equipment, and you go take a dive down in Chapter 5 of production, again, it talks about cross-contamination, that it should be prevented by attention to design. So you need to take account to your flows, the attention to your process design. Also an important aspect is when you look at another chapter of the Hydrolex is about quality control that explicitly says like that your microbiology laboratory should be arranged so as to minimize the risk of cross-contamination. So in other words, it should be segregated and separated from any other of your premises that you have to prevent that. And if you take a further look down into Chapter 5 about production, again, quality risk management, and that process, it should be included to assess the control of cross-contamination, including your facility equipment design and the use of it, your personnel and material flow. So although it's not very prescriptive saying like, okay, this is how you should do it, because of course every facility is unique and we talk about it in a bit more detail, it gives you guidance like, okay, this is what you need to take into account. So if you look back to the FDA guidance, it says about heating, ventilation, etc. And Hydrolex provides some additional information that you have to basically base your design based on the principles of quality risk management, so that you can justify what the risks are that you have identified in the design of your facility and how you're mitigating these by proper design of your facility. So if you then look at the Annex 1, which is specific for the manufacture of sterile medicinal products, or also applicable for products that are low bioburden or need some level of bioburden control, again, it emphasizes on processing and monitoring systems that need to be in place for the sterile product. Also, quality risk management priorities should be included to appropriate design of your facility. So although this Annex 1 came into effect in August 2023 and need to be in compliance, people need to be in compliance with this chapter since 2023, you have seen in previous sections of the Hydrolex that were effective since 2015 and after, it already spoke about that you have to apply quality risk management principles in the design of your facility. There are requirements for entry of personnel and material airlocks, specifics about the design and how to use these. There is this explicit mentioning of that direct human interventions. They need to be prevented in the critical zones by, for instance, using isolated technology, wraps, It talks about sealing designs, sinks and drains. And as you can see, there is a handy link below that people can refer to. So that's about the regulations. And now a bit of the practical aspects. And I would like to hand over back to my colleague, Gerardo.
Thanks, Patrick. So let's go to the more specifics about designing of a facility. Designing, for example, specifically on for aseptic processing, a clean room. This requires in-depth knowledge about many aspects, such as what type of products will be manufactured. Is it a liquid, a powder, or an ointment? I may add, is it a tablet or capsule? You know, what type of presentation and what type of product are you intending to make? If it's an injectable, or if it's not an injectable, then you're going into the second bullet there: sterile, non-sterile, or degree, or in between, low bioburden. We can go even further, like if it's sterile, if it's terminal sterilization, or if it's this aseptic processing, your facility design will be different. If it's high potency, that's a big one. Air flows are way different. Not only protecting the product, but protecting the people. Penicillin versus non-penicillin antibiotics, that has been around for quite a while. So everything for a penicillin facility must be totally separate, completely separate. Airflow, people, even laundry, everything totally different. And for the drug substance part, what type of molecule are we talking about? What type of protein are we manufacturing? If it's prokaryotic, mammalian, viral operations, that creates another element there. We can also see and talk about design of the facility based on the technologies that will be used. Are they open? Are they closed? Are they single use? Single-use facilities have been around trending in the last several years, mainly because of the type of products and the patient population. So single-use facilities, highly specialized, usually less of a square footage, but highly specialized and the same challenges as big facilities. Moving on to the next slide. Patrick, I think if you want to take this one, we can talk about that in comment.
Yeah, no, that's fine, Gerardo. Thanks for that. And as we have seen, and what is emphasized within both regulations as well, FDA and the European regulations, it is about flows, determine and based on your flows, how you lay out your facility. So you look at the flow diagrams of your product, of your personnel, your incoming materials, your sample flow, your waste flow, etc. All these aspects, you have to bring them together to come to an optimal design of your facility layout. Based on that, you can identify potential ingress points and take mitigation actions, say by using closed systems, adding in additional airlocks, etc. And then, of course, your HVAC design to take into consideration, like, are you using a recirculating air facility? As Gerardo said, like if you have a multi-product facility or a penicillin facility, that will be very difficult. Or are you availing of single-use air? Your pressure regime, you have to take into account the physical conditions, what is relevant to your product, but also, of course, for your personnel, about temperature, relative humidity. So it's very difficult to come to the right concept. And that's why these regulations can't be too prescriptive, because each manufacturing process, it is unique. And that's why you have to stand over why your design of your facility works as you have designed it. So having spoken about that, we have designed our facility now. So we want to move now to the qualification and commissioning. So Gerardo, would you take this away?
Absolutely. Thanks, Patrick. So qualification and commissioning, the three letters there are typically validation, qualification, and commissioning, or saying in the reverse order, commissioning, qualification, and validation, CQV. So very, very important and regulatory expectation. That's also in the regulation. From the FDA perspective, they talk about at the level of the law, they talk implicitly and sometimes explicitly using the word validation. But the general expectation is that you test your facilities for their intended use, your utilities, your equipment, and your systems. So you apply CQV to facilities, utilities, systems, equipment, and validation of your processes. We call that FUSEP for the first letters of each word. At the level of guidance, that's first bullet is the key one. The guidance for industry and process validation has been around since 2011, and the way this guide frame is in three stages: stage 1, stage 2, and stage 3. Stage 1 being process design, typically done at the process engineering level, high level R&D, that area of how you design your process. In stage 2 is where your typical commissioning, qualification, and validation comes in. That's stage 2. Then we internally refer sometimes like stage 2.1 and 2.2, meaning design of the facility, which is the first part of that. And then stage 2.2 is your PPQ, your traditional 3 batches of process validation towards the end of that. Then stage 3 is how do you maintain your validated state for everything, for facilities, utilities, equipment, and processes. So that stage 2.1 specifically is what typically we refer as the V model for validation. So your design of your facility and qualification, they explicitly talk about commissioning as well. So you apply commissioning to facilities and to many buildings, but you apply qualification and validation to equipment, systems, and processes. That is just another way of referring to that with a different V model. So I'll just jump to the similar concept, but from a European perspective. So back to you, Patrick.
Yeah, thanks, Gerardo. And yeah, Userlex Volume 4, Annex 15, it talks specifically about qualification and validation. And it is pretty much in sync what the FDA guidelines are describing, and also talk about, say, the V model that just was elaborated by Gerardo. How you start with your URS and with your the different stages your system is built, and then go into your installation qualification, operation qualification, and performance qualification, and how these are interlinked with each other. That basically your performance qualification checks your URS, etc., etc. And the next slide, it continues to build on that, how these interlinks are happening, and it also discusses the requalification of your facilities, your utilities, your systems, and your equipment, or as said, like fuse. So very important to follow this V model as well when you are qualifying your facilities, and to keep it in a state of control and in a validated state. So that brings us to some takeaway points at the end of our session here. As we have seen, the regulations, they are very broad. They give you general guidelines how to design your facility, what to take into account for. But how to do that, that is up to you. And for that, you need to know your product and your processes, what you're talking about. For that, use a cross-functional team. We see it so many times that it's from an engineering perspective, it's perfectly okay. But the end users and the people who are actually going to use the facility in the end, they are introduced and included at the latter stages, but they have to use the facility in the end. So use a cross-functional team in a timely manner, and let them have their seat at the table to discuss it. Apply the principles of quality risk management. This will allow you to identify potential issues, potential risks in advance that you can address in the design of your facility. And then commissioning, qualification, and validation of your fuel step using the product impact assessment and the quality risk principles of quality risk management. So that brings us to the end of this session. So I would like to ask if there are any questions for Gerardo or myself.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speakers now. So our first question is it expected that an airlock is always at low pressure compared to the higher classified area it leads to?
Gerardo, would you like to answer that one?
I will certainly do. The question, the answer is, it depends on the design. Typically, the airlocks are often at a higher pressure compared to the main hallway, but it depends on the type of design. And we talked about high potency drug products, so sometimes they are at a lower pressure compared to, so it acts as a sink. So it really depends on the design of your facility. Most often the time is a cascade for the traditional operations, but not necessarily.
All right, and our next question is, for clean room qualification, are airflow visualization studies required for areas with classification lower than grade A or B?
Yeah, I can answer that if you want. The simple answer is yes, especially when you manufacture sterile products or products that require some level of bioburden control. It must be demonstrated basically that there is no ingress from lower classified areas to your higher classified areas. This is now explicitly mentioned in the new or the current Annex 1, I must say, that became into effective in 2023 in August. So the information of air visualization studies is also to be considered when developing your EM program as per the same Annex 1. So it's basically twofold. It has a twofold function. But yeah, the simple answer is yes for that.
All right, and it looks like we're running a bit short on time. So if we don't get to your question, we'll be passing them along to Patrick and Gerardo, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is it a requirement to assess the recovery capability of a clean room when the conditions are lost?
I can take that one. It is an expectation it is a general expectation as part of your pressure, keeping your pressure differentials and protecting the product. The specifics of how to do it is more a best industry practice. But the short answer is yes, is an expectation. The how to or the specifics, it really varies. So you're more than welcome to reach out to us, either Patrick or myself, to provide more details towards that specific. That has a large Part B answer to that. So yes, it is a general expectation.
All right. Well, thank you so much, Patrick and Gerardo.
You're very welcome. Thanks. Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.